The Hidden Lives of Fungi, a gentle exploration of the Earth's quiet network, a sleep meditation and educational journey. Segment 1, The Kingdom Below. Welcome to the hidden world of fungi. In this series, we'll be exploring one of nature's most fascinating and least understood kingdoms, a world that exists all around us, beneath our feet, and in spaces too small for our eyes to see. This is a journey into the quiet lives of creatures that have shaped the planet in ways we're only beginning to understand. Let us begin at the very foundation of what it means to be a fungus. Many people wonder about the true nature of fungi, whether they belong with plants or animals or something else entirely. The answer is beautifully simple and profound. Fungi are not plants. They are not animals either. Fungi form their own kingdom of life, distinct and separate from all others, a testament to the diversity and creativity of evolution itself. This kingdom has existed for hundreds of millions of years, evolving in parallel to the plants and animals we know so well, yet remaining largely invisible to our everyday awareness. Now, let's explore what makes up this hidden world. The main body of a fungus is called the mycelium. Imagine, if you will, a vast network of delicate threads spreading through the soil, through wood, through countless substrates in our world. This is the mycelium. It is not a single, discrete organism, but rather a vast, interconnected web of filaments, each one working in concert with its neighbors. These individual threads are called hyphae, and they are where we find the true essence of fungal life. These hyphae are microscopic filaments, incredibly slender and delicate. They typically measure only 1 to 30 micrometers in diameter. To put this in perspective, a human hair is about 70 micrometers wide. So these fungal threads, these hyphae, are often thinner than a human hair, sometimes far more delicate. Yet despite their apparent fragility, they are remarkably strong and capable of extraordinary feats. But size can be deceiving, and here is where we encounter something truly remarkable. The mycelium, composed of these minute threads, can grow meters long. A single mycelial network might stretch across an entire forest floor, underneath a field, or through rotting wood. The total length of hyphae in a single cubic meter of soil might be measured in kilometers. This is the nature of the mycelium, individually invisible, collectively immense. In fact, the mycelial network is one of the largest living organisms on Earth. When we speak of the largest organisms on the planet, we often think of blue whales or the largest trees. Yet many fungal genets, which are individual fungal organisms, span vast areas of land. Their individual hyphae are too small to see, yet their collective presence is enormous, covering hectares of land, sometimes entire mountainsides. So how does this incredible network grow? Mycelium grows by a process called apical tip expansion. At the very tip of each hypha, growth occurs, with new cells being added and the filament extending forward into unexplored territory. This is a relentless, steady growth process. It is not chaotic or random, but guided by chemical signals, by the availability of nutrients, by the obstacles and opportunities in the environment. As the mycelium grows and expands, something remarkable happens. Hyphae branch and fuse. They split into multiple threads, creating new pathways for growth and resource distribution. They also merge together, fusing with compatible hyphae, creating connections and networks that become stronger and more resilient. This branching and fusing creates a structure that resembles something almost fractal-like, a tree-like network that is both organized and beautifully chaotic in its complexity. The network's density and branching patterns are not random or fixed. They are shaped by the conditions of life, nutrition availability in the environment, the ambient temperature, the moisture content, the presence of obstacles and opportunities. All these factors influence how the mycelium grows. A mycelium growing through nutrient-rich soil will develop differently than one growing through poor substrate. The network adapts, responsive and sensitive to its surroundings. Finally, within this delicate structure lies an intricate mechanics that governs how the network functions and behaves. The mechanics of mycelium are controlled by the elasticity of the individual filaments and by the architecture of their branching patterns. Each connection point, each junction where hyphae meet and fuse, plays a role in the overall strength and flexibility of the system. The network is not rigid, but rather flexible and dynamic, capable of adjusting to stresses and strains that might come from the environment or from the work of breaking down and transporting materials through the fungal body. Segment 2: The Architecture of Life. As we descend deeper into understanding the fungal network, we find ourselves contemplating the structure and function of this remarkable system. Let's begin with something beautiful and elegant: the purpose that the network structure serves. The network structure of mycelium allows for efficient resource transport across vast distances and provides resilience against damage and disruption. When a part of the network is damaged or destroyed, other pathways can compensate. When resources are abundant in one location and scarce in another, the mycelium can transport those materials along its highways of hyphae, distributing them throughout the organism. This is an engineering marvel designed by millions of years of evolution. One of the most fascinating discoveries about mycelium is that it can mimic the mechanical behavior of open-cell foam. If you have ever held a piece of foam in your hand, felt its springy, yielding quality, noticed how it compresses and expands, you have an intuition for how mycelium behaves under stress. This is not coincidental. The structure of hyphae, their branching patterns, their connections, create a material with properties similar to foam. This allows the mycelium to absorb stresses from its environment, to yield and flex without breaking, to distribute loads across the entire network. This mechanical behavior is quite remarkable when examined closely. The network exhibits nonlinear stress-strain behavior under both tension and compression. In simpler terms, this means that when you push on the mycelium with increasing force, it doesn't respond in a simple, linear way. Instead, the relationship between the force applied and the deformation produced changes as the stress increases. At first, the network might be quite springy, absorbing energy easily. As stress increases, the network stiffens and resists more. This complex response allows the mycelium to handle both gentle, ongoing stresses and sudden, intense pressures. Beyond this, mycelium exhibits something called strain hardening. This means that after the network has been deformed and stressed, it becomes stronger in response to that stress. Rather than being weakened by deformation, the mycelium adapts and hardens, preparing itself for future stresses. Additionally, the network exhibits hysteresis, meaning that the way it reacts to being compressed is different from the way it reacts to being released. There is a lag, a memory in the system, allowing the mycelium to store energy and respond dynamically to changing conditions. When we examine the strain distribution within the mycelium, we discover something quite sophisticated. The distribution is heterogeneous. This means it's not uniform throughout. Rather, there are regions of greater stress and regions of lesser stress. The network is sensitive enough that some sites within it are weaker than others, and these weaker sites are where collapse bands form when the mycelium is under significant stress. The network knows, in a sense, where it is most vulnerable, and it responds by concentrating strain in these locations while protecting more critical pathways. The coordination number of the mycelial network, meaning the average number of hyphae that meet at each connection point or node, is typically 4. This is a consistent feature across many fungal species. 4 connections create a balanced, stable architecture, allowing for good distribution of forces and efficient resource flow while maintaining flexibility. This is not arbitrary. Evolution has settled on 4 because this number provides an optimal balance of strength, flexibility, and efficiency. Network density is another important characteristic that we can measure and understand. Network density is defined as the total length of filaments per unit volume. In other words, if we take a cubic centimeter of soil colonized by mycelium and measure the total length of all the hyphae within it, we have measured the network density. This density varies depending on the fungal species, the age of the colony, the availability of nutrients, and many other factors. But understanding network density helps us appreciate the incredible amount of fungal material that can be present in even a small volume of soil. Now let's look at what makes up the physical structure of these hyphae. The walls of hyphae contain chitin, beta-glucans, and proteins. Chitin is a remarkable compound, the same substance that makes up the exoskeletons of insects and the shells of crustaceans. In the fungal cell wall, chitin provides rigidity and strength, giving the hyphae their structural integrity. Without chitin, the hyphae would be soft and weak, unable to maintain their form. Complementing the chitin is beta-glucan, which serves primarily as a mucilage on the outer surface of the hyphae. This slimy coating is not a weakness, but rather a sophisticated feature. The beta-glucan helps the hyphae interact with their environment, facilitates nutrient absorption, helps the mycelium navigate through soil, and creates an interface between the fungus and the broader ecosystem. It is a chemical language, a way for the fungus to speak to the soil, to microorganisms, to plant roots, to the world around it. Finally, we must consider that mycelium is statistically isotropic. This elegant term means that the structure of the mycelium is uniform in all directions. While individual hyphae grow in response to stimuli and may orient themselves in particular directions, when we look at the overall network structure, it appears the same whether you view it from above, below, or from any angle. This uniformity is an advantage, providing equal strength and equal resource distribution capabilities in all directions. Segment 3: Resilience and Adaptation. One of the most striking features of mycelial networks is their capacity to adapt and recover from adversity. Let's explore the remarkable ways in which fungi respond to the challenges of their environment. First, we recognize that the network can reorganize itself in response to environmental changes. The mycelium is not static or fixed in its form. Rather, it is a living system that senses its environment continuously and adjusts accordingly. If conditions change, if new nutrients become available in one direction, if an obstacle appears in another, the mycelium alters its growth pattern, redirects its expansion, and reorganizes its architecture. When mycelium experiences mechanical stress, it responds in a particularly interesting way. Mycelium responds to mechanical stress by forming multiple collapse bands. Rather than failing catastrophically across its entire structure, the mycelium concentrates the deformation into specific regions, what we call collapse bands. These are areas where the network structure temporarily fails or collapses under stress, allowing the rest of the network to remain intact and functional. This is a sophisticated strategy for managing damage and preserving the organism's viability. What's particularly remarkable is that this response leads to local network reorganization. After stress has occurred and collapse bands have formed, the local structure of the network changes. The hyphae reorganize, new connections form, and the architecture is restructured. This reorganization has interesting consequences. The reloading curves, which describe how the network responds to stress in subsequent loading events, are dramatically different from the initial response. The mycelium has learned, adapted, and prepared itself for future stress based on past experience. This resilience extends to the mycelium's capacity to recover from deformation and adapt its structure. After deformation occurs, the mycelium can reverse the damage, returning to its normal form. But more than that, it adapts its structure in response to the experience. The mycelium becomes stronger, better organized, more prepared for future challenges. This is a form of memory, a way in which the fungus learns from its past and improves its future resilience. The network's topology, which is the overall shape and organization of its structure, is shaped by the nutritional and environmental conditions it experiences. Topology is not predetermined or fixed at the moment of spore germination. Instead, it emerges dynamically from the ongoing interaction between the fungal organism and its environment. In nutrient-rich conditions, the mycelium might develop one topology. In nutrient-poor conditions, it might develop quite differently. In areas with plenty of moisture, it might organize itself differently than in dry regions. The mycelium is a sculptor of itself, shaping its own form in response to the world around it. Now let's consider the sheer scale that some mycelial networks can achieve. Mycelium can form large, interconnected colonies spanning hectares of land, multiple hectares, covering the area of many football fields, all part of a single, connected fungal organism. These are not separate fungi that happen to be in the same location, but rather individual organisms of enormous size. Imagine walking across a field or through a forest, and the ground beneath your feet is all part of a single living creature, a single fungal organism. The largest known fungal genet, which is a term for an individual fungal organism, covers an astonishing 965 hectares of land. This is located in the Malheur National Forest in Oregon, United States. This honey fungus, a species called Armillaria ostoyae, is believed to be thousands of years old. It's estimated to weigh hundreds of tons. For thousands of years, this single organism has been growing, expanding, breaking down wood, feeding the forest ecosystem, a truly ancient and monumental presence. Within these vast networks, we find specialized structures called rhizomorphs. Rhizomorphs are specialized root-like structures that extend through soil, sometimes above the soil surface, sometimes deep underground. These structures are more organized and compact than the diffuse mycelium, resembling roots in their form and function. The rhizomorph has a central cavity or lacuna, surrounded by a thick, melanized rind, a dark outer coating that provides protection and strength. These rhizomorphs act as high conductivity channels for nutrient transport. They are, in essence, the highways of the fungal network, specialized structures built for rapid and efficient transport of water, nutrients, and other materials across long distances. A fungus might use its diffuse mycelium for general exploration and nutrient acquisition, but when it needs to move significant quantities of material from one part of its colony to another, it uses the rhizomorphs. This is another example of the sophistication of fungal organization. Finally, within a single mycelial network, we find that mycelium can differentiate into distinct zones with different functions. Different regions of the same organism may be specialized for different tasks. One zone might be focused on exploring new territory and acquiring nutrients. Another might be focused on storing energy and resources. A third might be involved in reproduction or in communication with plant roots or other organisms. The mycelium is not uniform in function, but rather a specialized, multifaceted organism capable of maintaining diverse roles simultaneously. Segment 4, the magic of reproduction. Now we turn our attention to one of the most fundamental aspects of fungal life, reproduction. Through reproduction, fungi ensure their survival and spread their influence across the world. Let's begin with the foundational concept. Mycelial networks are essential for fungal reproduction. Without the mycelium, without this vast interconnected network of hyphae, fungi could not reproduce. The mycelium is not merely a feeding structure or a nutrient transport system. It is the foundation upon which all reproduction is built. When we speak of fungal reproduction, we must understand something quite remarkable about compatibility and fusion. Compatible mycelia can fuse together, allowing DNA mixing and genetic exchange. When two mycelial networks of compatible mating types come into contact, something remarkable happens. They recognize each other chemically at the cellular level. The barriers between them dissolve and they merge, their cytoplasm mixing, their genetic material mingling. This is the fungal equivalent of sexual reproduction, though it operates quite differently from the reproduction we see in animals and plants. Once fusion has occurred, once compatible mycelia have merged, cells within the network combine and form new spores. Spores are remarkable structures, tiny packages of genetic material and cellular machinery capable of surviving harsh conditions and eventually germinating into new fungal organisms. These spores are the ambassadors of the fungus. capable of traveling great distances through air, water, or the digestive systems of animals, spreading the fungal species far and wide. The location of these spores varies depending on the fungal species. Some fungi hold spores inside protective structures called truffles, fruiting bodies that develop underground, surrounded by protective tissue. Other fungi expose their spores in mushrooms, the visible fruiting bodies that we see popping up in forests and fields after rain. Whether hidden underground or displayed in the air, these spore-bearing structures are the result of the mycelial network's reproductive efforts, the visible manifestation of processes that have been occurring in the darkness beneath our feet. At a deeper level, mycelium plays a crucial role in fungal sexual reproduction. The mycelium is not merely a passive structure that hosts reproduction. Rather, it actively participates in the reproductive process, organizing the cellular machinery, directing resources toward the developing fruiting bodies, and managing the complex biochemistry that leads to spore production. The mycelium is both the parent and the midwife of the next generation. Extending this understanding, we recognize that the network allows fungi to reproduce sexually when conditions are right. Fungi have the capacity to sense environmental conditions, the temperature, the moisture level, the availability of nutrients, the presence of other organisms. When conditions are favorable for reproduction, when the mycelium has accumulated sufficient resources and environmental conditions are optimal, the fungus can initiate sexual reproduction. It directs energy and materials from throughout the network toward fruiting body development. This is a coordinated, intelligent response to environmental conditions, a decision made at the level of the entire organism. Now we shift our perspective to consider one of the most beautiful and consequential relationships in nature: the connection between fungi and plants. Mycelium can connect individual plants, forming what we might think of as a web of relationships between organisms. These connections are not accident or coincidence, but rather the result of specialized fungal associations with plant roots called mycorrhizal relationships. Mycorrhizal networks transfer water, nitrogen, and carbon between plants. Through the mycelium, these essential resources flow between plant individuals. A plant in a nutrient-rich patch of soil producing excess nitrogen can transfer that nitrogen through the mycorrhizal network to a plant in a nutrient-poor patch. In the process of photosynthesis, plants produce sugars and carbon compounds. Some of these are transported through the mycorrhizal network to feed the fungus and potentially to feed other plants. Water absorbed by the mycelium can be delivered to plants during dry periods. The network is a system of exchange and mutual support. This interconnected plant community, linked together by fungal networks, is often called the Wood Wide Web. The term is used somewhat poetically to describe the vast, interconnected nature of fungal networks in forests and woodlands, linking trees and other plants together in a vast, living internet of fungal threads. This metaphor is not merely cute. It captures something true about the functional reality of these networks. Information flows through them. Resources flow through them. The forest is connected, unified by fungal networks. Mycelium wraps around plant roots, forming symbiotic relationships where both parties benefit. The fungus receives sugars produced by the plant through photosynthesis. In return, the plant receives access to water and minerals from the soil, accessed by the fungus with its vast network of hyphae. Neither partner could thrive as well alone. Together, they are stronger. The network acts as an exchange medium for nutrients and growth-promoting materials. The mycelium doesn't merely transport resources, it processes them, transforms them, makes them available in forms that both the fungus and its plant partners can use. Certain minerals, like phosphorus, are bound up in soil in forms that plant roots cannot access directly. But the fungal mycelium can break these minerals loose, transform them into available forms, and deliver them to the plant. In return, the plant provides the fungus with carbohydrates and other organic compounds that the fungus needs. Beyond these material exchanges, we find something even more remarkable. Mycelium can produce electrical gradients for electrophysiological signals. This means that the mycelium generates differences in electrical charge across its structure, creating the physical substrate for sending signals and receiving information. These electrical signals may carry information about nutrient availability, about the presence of pathogens or predators, about the overall state of the mycelial network. This is a form of nervous system, fungal cognition occurring at the cellular level. Segment 5. The language of plants and fungi. The relationship between fungi and plants goes beyond simple material exchange. There is communication, recognition, and sophisticated coordination between these two kingdoms of life. Let's explore the ways in which plants and fungi speak to each other in the darkness of the soil. Plants tend to root towards fungi that supply more nutrients. This might sound surprising. How can a plant distinguish between different fungal partners? The answer lies in the plant's sensitivity to its own nutrient status. A plant that is receiving abundant nutrients from its fungal partners grows vigorously, develops more resources, and expands its roots in the direction of this abundance. Conversely, a plant that is receiving fewer nutrients from its fungal associations may expand its roots in different directions, seeking new fungal partners or other sources of nutrients. The plant, through its behavior, is responding to the quality of its fungal relationships. Mycelium helps plants access essential minerals like phosphorus and nitrogen. Phosphorus is often one of the most limiting nutrients in natural soils, locked away in forms that plant roots cannot access. Yet mycelium, with its vast surface area and sophisticated chemistry, can break phosphorus free from soil minerals and deliver it to plants. Nitrogen, another critical nutrient, can be captured by specialized fungi working together with bacteria, then delivered to plants. Without these fungal helpers, many plants would struggle to obtain sufficient nutrients, regardless of how rich the soil might seem. Now, we expand our view to consider the role of fungi in the broader ecosystem. The network is crucial for nutrient cycling in ecosystems. Ecosystems depend on cycling, the continuous movement of nutrients from the soil to living organisms and back again. Without this cycling, nutrients would accumulate in death and decay, becoming unavailable for new growth. Fungi, through their mycelial networks, are the primary drivers of this cycling. At the foundation of this process, mycelium decomposes organic matter, recycling nutrients. When a tree falls, when leaves drop in autumn, when any organism dies, that material must be broken down and returned to the soil. Fungi are the primary decomposers in terrestrial ecosystems. The mycelium extends through dead wood, through fallen leaves, through soil rich in organic matter. The fungal enzymes break down complex compounds, transforming them into simpler forms that can be reused by living organisms. This is not a fast process, but a slow, steady work that has continued for hundreds of millions of years. Through this decomposition, fungi play a key role in soil formation and fertility. Soil is not just broken down rock and dead organic matter. It is a living system, and fungi are central to its development and maintenance. As mycelium decomposes organic matter, it integrates that organic material into the soil. It stabilizes soil structure, improving water retention and nutrient availability. Fungal metabolites enrich the soil, supporting the growth of bacteria and other microorganisms that further enhance soil fertility. Over time, through the combined action of fungi, bacteria, plants, and countless other organisms, fungal-rich soil becomes deep, dark, fertile, capable of supporting rich ecosystems. This decomposition capability extends to complex organic compounds. Mycelium can break down complex organic compounds that many other organisms cannot touch. Lignin, the tough compound that gives wood its strength, can be broken down by certain fungi. Cellulose, the primary structural component of plant cell walls, is broken down by fungal enzymes. Chitin, the same compound that makes up their own cell walls, can be broken down by fungi. These decomposition capabilities are crucial for nutrient cycling on a global scale. Beyond decomposition and nutrient cycling, fungi play a role in carbon sequestration. The network is involved in carbon sequestration, the process of capturing and storing carbon in soil. As mycelium breaks down organic matter, some of the carbon is released as carbon dioxide into the atmosphere. But some of this carbon is incorporated into fungal biomass, into the bodies of fungi themselves. When these fungi die, their carbon-rich bodies accumulate in soil, locked away from the atmosphere. Over time, fungal accumulation in soil represents a significant sink for atmospheric carbon, a natural process that helps regulate Earth's climate. Mycelium can transport resources across large distances, allowing the fungus to exploit its environment efficiently. The network allows fungi to forage for nutrients in patchy environments. In nature, nutrients are rarely evenly distributed. There might be abundant nutrients near a fallen log or beneath an animal carcass, while other areas are relatively nutrient poor. The mycelium, with its ability to extend over large areas, can locate these nutrient-rich patches and exploit them. The resources acquired in these patches can then be transported throughout the network, supporting the fungus in areas where resources are more limited. Segment 6: Intelligence, Communication, and Community. As we contemplate the fungal network further, we begin to recognize that it is not merely a passive structure for resource transport and nutrient acquisition. It is a living system with properties that we might even describe as intelligent or communicative. Let's explore these remarkable aspects of fungal life. Mycelium can respond to competition and predation. The mycelial network is not isolated from other organisms. Rather, it coexists with bacteria, nematodes, insects, plant roots, and countless other life forms, competing for resources, predation, and being preyed upon. The mycelium can sense these challenges. When a competing fungus invades territory, the mycelium can respond, producing compounds that inhibit the competitor. When a nematode begins to feed on hyphae, the mycelium can respond, producing compounds that trap or poison the nematode. These are not conscious decisions, but rather sophisticated biochemical responses to threats and challenges. When the mycelium is damaged, it can repair and regenerate damaged sections. A hypha broken by a passing animal can be bypassed. A section of the network isolated by a competing fungus can be lost while the rest of the network survives and thrives. The mycelium is resilient, capable of adjusting to damage and loss, maintaining its overall function and growth. At the core of these capabilities lies something fundamental. Mycelium can form mutualistic relationships with other organisms. A mutualistic relationship is one where both parties benefit. The mycorrhizal relationships we discussed earlier are mutualistic, but fungi form mutualistic relationships far beyond plants. Bacteria that live on the surface of hyphae may protect the fungus from certain pathogens while receiving carbohydrates from the fungus. Small animals that feed on fungal fruiting bodies may spread fungal spores while obtaining nutrition. The fungus exists not in isolation, but as a member of a community, engaging in relationships of mutual benefit. The network is essential for fungal communication. This might sound strange. How do fungi communicate? Through chemistry, through the movement of signals and information throughout the mycelial network. A mycelial network extends through a three-dimensional space, and information can be transmitted from one region to another. If nutrients are discovered in one area, signals can broadcast this information throughout the network. If a threat is detected, warning signals can be transmitted. If fruiting conditions are ideal, the entire network can be coordinated toward reproductive effort. This is communication, not in the sense of language and words, but in the sense of information transmission and coordinated response. Mycelium can sense and respond to environmental cues. The mycelium is not blind to its environment. Rather, it continuously senses conditions around it and responds accordingly. Temperature, moisture, light, if near the surface, the presence of certain chemicals, the vibrations of movement nearby, the touch of another organism, all these stimuli are detected and responded to by the mycelium. This sensory capability, distributed throughout the entire network, gives the fungus a sophisticated awareness of its environment. It can alter its growth pattern in response to resources. When resources are abundant in one direction, the mycelium can redirect its growth toward that abundance. When a source of nutrients becomes available, the mycelium can orient its growth toward it. This is not by accident or random chance, but by directed response to environmental cues. The mycelium is not passively waiting for nutrients to come to it. It is actively seeking them out, guided by chemical signals and other cues that indicate where resources are most abundant. The diversity of mycelial forms is remarkable. Mycelium can form dense mats or sparse networks depending on environmental conditions. In nutrient-rich substrates with high moisture, mycelium might form dense mats, thickly woven networks with high fall density. In nutrient-poor areas or in dry conditions, the same fungal species might form more sparse, extended networks designed to cover maximum territory with minimum resources. The network structure is highly plastic and adaptable, changing in response to conditions and being reshaped by the fungus's current needs and challenges. Beyond its relationship with plants, fungi form symbiotic relationships with other photosynthetic organisms. Mycelium can form symbiotic relationships with algae and cyanobacteria. In these partnerships, the fungus provides structural support and protection, while the algae or cyanobacteria provide carbohydrates through photosynthesis. These relationships can form lichens, which are composite organisms consisting of fungal mycelium and photosynthetic partners, forming a single integrated organism. Lichens are remarkable in their ability to colonize harsh environments, growing on bare rock, on tree bark, in deserts and Arctic regions, living evidence of the power of fungal partnerships. Mycelium can colonize extreme environments. The mycelium is not restricted to mild, comfortable conditions. Fungi can be found in deserts, in Arctic tundra, in caves, in hot springs, in deep ocean sediments. The mycelium, flexible and adaptable, can adjust to extraordinary conditions that would kill most organisms. The network can survive in harsh conditions, maintaining its functions even when exposed to temperature extremes, desiccation, or high levels of radiation. Segment 7. Fungal communities and interactions. The hidden world of fungi is not one where individual organisms live in isolation. Rather, it is a world of communities, of complex interactions between fungi and the vast array of microorganisms and organisms with which they coexist. Let's explore this rich microbial and organismal landscape. Mycelium can form biofilms on surfaces. A biofilm is a community of microorganisms embedded in a protective matrix. Many people are familiar with biofilms through dental plaque or algae slime on rocks, but fungal biofilms are equally important. When mycelium grows on a surface, it can form a biofilm, a structured community that may include fungal hyphae, bacteria, and other microorganisms. This biofilm matrix provides protection and enables sophisticated interactions between community members. It can interact with bacteria and other microbes. The relationship between fungi and bacteria is complex and multifaceted. In some cases, bacteria break down compounds that the fungus cannot, making those compounds available for the fungus. In other cases, fungi produce compounds that inhibit bacterial growth, controlling bacterial populations. In still other cases, fungi and bacteria work together, their activities complementary, supporting each other's survival and function. The mycelium exists in a microbial milieu, constantly interacting with the bacterial world around it. Mycelium can form biofilms that protect against environmental stress. The protective matrix of a fungal biofilm can shield the organisms within it from harsh environmental conditions. The matrix can retain moisture, protecting against desiccation. It can insulate against temperature extremes. It can buffer against harsh chemicals or UV radiation. For an organism living in an environment exposed to environmental stress, a biofilm is a refuge, a protective community that enables survival in conditions that would be lethal to an isolated cell. The network can facilitate horizontal gene transfer. This is a sophisticated and important process. Horizontal gene transfer is the movement of genetic material between organisms, particularly between members of different species. This is different from vertical gene transfer, where genetic material is passed from parent to offspring. In biofilms and other close associations, fungi can exchange genetic material with bacteria and potentially with other fungi. This allows the fungus to acquire useful genes, traits that might provide advantages in its particular environment. Over time, this horizontal gene transfer has allowed fungi to acquire genes enabling them to break down novel compounds, resist antibiotics, or tolerate harsh conditions. Mycelium can form complex communities with other organisms. Life in nature consists of complex communities, webs of relationships between multiple species. The mycelium is typically at the center of these communities, providing structure, exchanging resources, facilitating interactions between community members. A cubic centimeter of soil might contain fungal hyphae, bacteria, protozoa, nematodes, tiny arthropods, all interacting in complex ways, all supported by the fungal infrastructure. It can form mutualistic relationships with insects. Many insects depend on fungi, and many fungi have evolved to support insect populations. Some ants farm fungi, cultivating them as a food source, protecting the fungi and spreading them to new locations. Some insects feed on fungal fruiting bodies and in the process spread fungal spores. Certain fungal species have evolved fruiting bodies that are attractive to insects, brightly colored and fragrant, advertising their presence in the same way that flowering plants advertise to pollinators. Insects and fungi have co-evolved, developing interdependencies that bind them together. Mycelium can form symbiotic relationships with animals. Beyond insects, fungi form relationships with a diversity of animals. Mammals, birds, amphibians, and reptiles all interact with fungi. Some animals deliberately consume fungal fruiting bodies, obtaining nutrition from the fungus. In the process, they disperse fungal spores, acting as fungal gardeners spreading the fungus across vast distances. Other relationships are more subtle, with animals being affected by fungi in ways they don't consciously recognize. The network can interact with the immune system of animals. Fungi are not entirely outside the animal body. Fungi can colonize animal skin, animal respiratory systems, animal digestive systems. In these interactions, the animal immune system must distinguish between potentially harmful fungi and beneficial or commensal fungi. The immune system has evolved sophisticated mechanisms to respond appropriately. Too weak a response and potentially pathogenic fungi might proliferate. Too strong a response and beneficial fungi might be eliminated. This ongoing negotiation between fungal networks and animal immunity is a constant dance, a relationship of power and restraint. Mycelium can form biofilms in animal hosts. When fungi establish themselves within animal hosts, they may form biofilms, structured communities that enable their survival and persistence within the host. These biofilms can be resistant to immune attack and to antifungal medications, making them particularly challenging to treat when they become pathogenic. Yet in many cases, these fungal biofilms are benign or beneficial, part of the normal microbiome of the animal host. Segment 8: Aquatic Worlds and Diversity. So far, we have focused much of our attention on terrestrial fungal networks, but fungi extend their domain into aquatic environments as well. Let's explore how fungi thrive in marine and freshwater ecosystems, adapting to the unique challenges and opportunities that water presents. It can form mutualistic relationships with marine organisms. The ocean is not a fungi-free zone. Aquatic fungi exist in seawater, in freshwater, colonizing substrates, forming relationships with marine organisms. Many marine animals depend on fungi for decomposition, nutrient cycling, and food. Some marine organisms, in turn, provide environments for fungi or disperse fungal spores. These relationships are less visible to us than terrestrial fungal relationships, hidden beneath water, but no less important for the functioning of aquatic ecosystems. Mycelium can colonize aquatic environments. Fungi have evolved to live in water, despite the challenges that aquatic life presents. Water brings nutrients and creates challenges for osmotic regulation, yet fungi have adapted, evolving mechanisms to survive and thrive in freshwater lakes, rivers, and oceans. Aquatic fungi may form less extensive mycelial networks than terrestrial fungi, or may adapt their hyphal structure to deal with water currents and aquatic conditions. The network can form symbiotic relationships with marine plants. Kelps and seagrasses are important primary producers in marine ecosystems, yet they too form relationships with fungi. Fungal partners help marine plants obtain nutrients, protect them from pathogens, and support their growth. These partnerships are less well studied than terrestrial mycorrhizal relationships, but they are equally important for the functioning of marine ecosystems. Mycelium can form biofilms on marine surfaces. Submerged rocks, wood, and other surfaces become colonized by marine biofilms, communities that include bacteria, algae, and fungi. These biofilms are important components of aquatic ecosystems, processing organic matter, cycling nutrients, and providing habitat for small organisms. The fungal component of these biofilms plays a crucial role in decomposition and nutrient cycling in water. It can interact with marine bacteria and other microbes. The relationship between fungi and bacteria in marine environments is just as complex as in terrestrial environments, though perhaps less studied. Marine fungi interact with marine bacteria, forming relationships of competition, cooperation, and mutual benefit. These microbial communities are crucial for the cycling of nutrients in the ocean, for the breakdown of organic matter that falls from the upper ocean to the depths, and for the regulation of microbial populations. Mycelium can form mutualistic relationships with marine animals. From tiny zooplankton to whales, marine animals interact with fungi in various ways. Some filter-feeding organisms may consume fungal spores or hyphal fragments. Some animals may harbor fungal partners in their guts or on their skin. Others may depend on fungi for the decomposition of food items or the cycling of nutrients in their environment. These relationships are diverse and often subtle, but they contribute to the overall functioning of marine ecosystems. The network can interact with the immune system of marine animals. Just as with terrestrial animals, marine animals have immune systems that must contend with fungal colonization. Fish, marine mammals, and marine invertebrates all have evolved immune mechanisms to deal with fungal encounters. Some marine fungi may be pathogenic, while others are beneficial or neutral. The balance between these is constantly negotiated at the interface between fungus and host. Mycelium can form biofilms in marine animal hosts. When fungi establish themselves in marine animal hosts, they may form biofilms, just as they do in terrestrial animals. These biofilms in marine animal hosts can be beneficial, harmful, or neutral, depending on the specific fungal-host combination. Certain fungal diseases in marine organisms are important conservation concerns, affecting wild populations of fish and marine mammals. The network can facilitate nutrient cycling in marine ecosystems. Ultimately, the presence of fungi in marine ecosystems contributes to nutrient cycling, much as it does in terrestrial ecosystems. Fungi decompose organic matter, transform nutrients, make them available for uptake by living organisms, and help regulate the movement of carbon, nitrogen, and phosphorus through marine food webs and biogeochemical cycles. Mycelium can form symbiotic relationships with marine plants. We have mentioned this before, but it bears repeating because of its importance. Marine fungi forming relationships with kelps, seagrasses, and other aquatic plants contribute to the productivity and health of marine ecosystems. These partnerships may be less visible and perhaps less well studied than terrestrial mycorrhizal relationships, but they are real and significant components of marine ecology. Segment 9: The Fungal Foundation of Terrestrial Ecosystems. We now return our attention to terrestrial ecosystems and explore in greater depth the ways in which fungi form the foundation upon which terrestrial life depends. The relationships between fungi and terrestrial life are so intimate, so pervasive, that we might say terrestrial life could not exist without fungi. Mycelium can form symbiotic relationships with terrestrial plants. We have touched on this throughout our exploration, but it deserves to be returned to again and again because it is so fundamental. The vast majority of terrestrial plants form symbiotic relationships with fungi. These relationships are mutualistic, mutually beneficial. The plant provides sugars to the fungus, energy captured from the sun through photosynthesis. The fungus provides minerals and water to the plant, resources acquired through the mycelial network's exploration of the soil. Without these partnerships, terrestrial plant communities would be far less productive, far less diverse, far less able to thrive. The network can interact with the root system of plants. The mycelium doesn't merely wrap around plant roots, it enters into intimate communication with the root system. Fungal hyphae penetrate plant roots, forming connections at the cellular level. These connections are not parasitic, though it might seem that way. Rather, they are carefully regulated interactions where both partners benefit. The plant receives minerals and water. The fungus receives carbohydrates. The interaction is governed by chemical signals, by the selective opening and closing of pathways, by a finely tuned negotiation between fungal and plant partners. Mycelium can form mutualistic relationships with terrestrial animals. Beyond plants, fungi form relationships with terrestrial animals. Many terrestrial animals depend on fungal fruiting bodies for food. Many terrestrial animals help disperse fungal spores. Some terrestrial animals harbor fungal partners in their guts. Fungal communities that assist in digestion or nutrient absorption. The diversity of fungal-animal relationships in terrestrial ecosystems is remarkable and often overlooked. It can interact with the digestive system of animals. Some animals that consume plant material depend on fungi to help break down that material. The fungal enzymes that can break down cellulose and lignin are particularly valuable in the digestive systems of herbivores and omnivores. Some animals harbor fungi in their guts, fungal communities that are as important to digestion as the animal's own enzymes. When such animals consume fungal fruiting bodies or fungal spores, they may be deliberately cultivating fungal partners that assist in digestion. Mycelium can form symbiotic relationships with terrestrial insects. Insects and fungi have intertwined destinies. Many insects depend on fungi for food, either by consuming fungal fruiting bodies or by cultivating fungal gardens. Many fungi depend on insects for spore dispersal and pollination of sorts, with insects visiting fruiting bodies, consuming spores, and depositing them in new locations. Some insects and fungi have evolved highly specialized relationships, where each depends critically on the other. The network can interact with the immune system of terrestrial animals. Terrestrial animals encounter fungi constantly. The fungi on their skin, in their respiratory systems, in their digestive systems, all require immune attention. The immune systems of terrestrial animals have evolved sophisticated mechanisms to distinguish between beneficial, commensal, and pathogenic fungi. This is an ongoing negotiation, a balance between immune tolerance and immune defense. A relationship that is critical for the health and well-being of the terrestrial animal. Mycelium can form biofilms in terrestrial environments. The soil is full of biofilms, communities of fungi, bacteria, and other organisms embedded in protective matrices. These terrestrial biofilms are sites of intense activity, where nutrients are cycled, where organic matter is decomposed, where organisms interact and communicate. The terrestrial biofilm is where much of the essential work of the ecosystem occurs, in the dark, moist, nutrient-rich spaces just beneath the surface of the soil. It can interact with terrestrial bacteria and other microbes. The relationship between fungi and bacteria in terrestrial environments is perhaps more complex than any other microbial interaction. Fungi and bacteria compete for the same resources. They inhibit each other through the production of antibiotics and other compounds. Yet they also cooperate, with fungi breaking down compounds that bacteria cannot, and bacteria accessing nutrients that fungi cannot. In decomposition, in nutrient cycling, in the processing of organic matter, fungi and bacteria work together, their activities complementary and interconnected. Mycelium can form mutualistic relationships with terrestrial plants. This relationship, mentioned multiple times throughout our exploration, is so important that we return to it again. Mycorrhizal relationships between fungi and terrestrial plants are found in the vast majority of terrestrial plant species. These relationships are found in forests, in grasslands, in deserts, in Arctic tundra. They have evolved repeatedly, independently, in different fungal and plant lineages. This repetition and ubiquity speaks to the profound importance of mycorrhizal relationships for terrestrial plant life and terrestrial ecosystem functioning. The network can facilitate nutrient cycling in terrestrial ecosystems. In the end, the role of fungi in terrestrial ecosystems comes down to nutrient cycling. Fungi take the dead remains of organisms, the fallen leaves, the dead roots, the animal droppings, and they break these down, transform them, return them to forms that living organisms can use. Through this endless cycling, through this perpetual process of decomposition and nutrient release, fungi make terrestrial life possible. Without fungi, dead matter would accumulate, nutrients would be locked away, new life could not flourish. Fungi, through their mycelial networks, are the custodians and recyclers of terrestrial nutrients. Segment 10: Ancient Partnerships and Evolutionary History. We now take a step back from the detailed mechanisms of fungal life and consider the grand historical arc, the evolutionary history that has created the fungal kingdoms we see today. This history reveals ancient partnerships, evolutionary innovations, and the deep origins of fungal relationships with other forms of life. The origin of fungi stretches back nearly 1 billion years into Earth's past. Fungi diverged from animals at some point in the distant past, evolving their own distinct characteristics and strategies for life. The earliest fossils that can confidently be identified as fungal structures date back several 100 million years, though fungi likely originated earlier. From this distant past, fungi have evolved an extraordinary diversity of forms and lifestyles, adapting to nearly every environment on Earth. The development of mycorrhizal relationships, the partnerships between fungi and plant roots, occurred at a critical juncture in Earth's history. When plants first moved onto land, around 450 million years ago, they faced a fundamental challenge. The soil on land was not as easily accessible as the water in which plants had evolved. Land plants needed roots to anchor themselves and to access water and minerals from soil. But terrestrial soils were initially relatively poor in minerals. The transition from water to land would have been nearly impossible without fungal partnerships. Evidence suggests that the very first land plants had fungi associated with them, that mycorrhizal relationships may have made the colonization of land by plants possible. This ancient partnership has continued to evolve over hundreds of millions of years. Different groups of plants evolved specialized relationships with different groups of fungi. Orchids, for example, have evolved highly specialized mycorrhizal relationships where the fungus is absolutely essential for seed germination and early growth. Many trees in temperate forests depend on ectomycorrhizal fungi, where the fungal mycelium forms a sheath around the plant root rather than penetrating it. The extraordinary diversity of mycorrhizal relationships we see today is the product of millions of years of coevolution. The evolution of fungal fruiting bodies, the visible mushrooms and other spore-producing structures reflects adaptations to specific environments and dispersal opportunities. Some fungi have evolved fruiting bodies that are deliberately attractive to animals, fruiting bodies that smell pleasant, that are brightly colored, that provide nourishing food to the animals that consume them. In return, these animals disperse the fungal spores. Other fungi have evolved inconspicuous fruiting bodies, or produce spores in such quantities that the sheer numbers ensure some spores find their way to suitable environments. Still others produce spores that are adapted to wind dispersal, lightweight, and aerodynamic. The diversity of fungal decomposers reflects the complexity of organic matter and the evolutionary innovations fungi have developed to exploit different substrates. Some fungi have specialized in decomposing wood, some in decomposing leaves, some in decomposing cellulose, some in decomposing chitin, some in decomposing even more resistant compounds. Each of these adaptations required the evolution of specific enzymes, the ability to produce and secrete the right biochemical machinery to break down the target compound. This enzymatic innovation has been driven by millions of years of natural selection, with fungi that could decompose new substrates gaining access to new food sources and new opportunities for growth and reproduction. The relationships between fungi and animals have evolved extensively. Consider the ecology of a truffle, a fungus that produces its fruiting bodies underground. Truffles are highly aromatic, smelling strongly of distinctive compounds. These odors have been shaped by evolution to be attractive to mammals and other animals that can then dig up the truffles, consume them, and disperse the spores through their digestive systems. The animals are attracted to the truffle's scent, just as they are attracted to the scent of a mate or a food source. This is an example of coevolution, where the fungus has evolved characteristics that manipulate animal behavior for the fungus's benefit, while the animal has evolved sensory systems and behaviors that lead it to consume fruiting bodies that may actually provide nutrition. The evolution of fungal toxins and defensive compounds reflects the ongoing arms race between fungi and their enemies. Many fungi produce compounds that are toxic to bacteria, to insects, to grazing animals. These defensive compounds have evolved as specific responses to specific threats. A fungus that can produce an antibiotic that kills competing bacteria gains an advantage. A fungus that can produce a compound that repels insect feeding gains an advantage. Over evolutionary time, fungi have evolved an enormous arsenal of defensive compounds, many of which have proven useful to humans as medicines and pesticides. The evolution of fungal communication systems, including the production of chemical signals and electrical gradients, has allowed fungi to coordinate activities across their vast networks. A single fungal organism might span many meters or hectares. Coordinating the activities of this entire organism, directing resources toward fruiting body production when conditions are favorable, responding to threats, exploiting newly discovered food sources. All of this requires communication. The evolution of fungal communication systems has been a critical innovation, allowing fungi to effectively manage their enormous bodies. The radiation of fungal diversity following major extinction events demonstrates the flexibility and adaptability of fungi. When other organisms went extinct, when ecosystems were devastated, fungi often persisted. Some fungal species might go extinct, but the lineage survives, and in the aftermath of extinction, with reduced competition and new ecological opportunities, fungi evolve rapidly, diversifying to fill new ecological roles. This has happened multiple times throughout Earth's history, and each time, the diversity and dominance of fungi has increased. Segment 11. The mechanisms of fungal sensing and response. Having considered the historical arc of fungal evolution, we now delve into the mechanisms by which fungi sense their environment and respond to changes around them. These mechanisms, operating at the molecular and cellular level, provide fungi with their remarkable ability to adapt and survive. The mycelium senses chemical gradients in its environment. Fungi are exquisitely sensitive to chemical signals. A hypha can detect the presence of a nutrient source at a distance and grow toward it. How is this possible? Through the detection of chemical gradients, differences in the concentration of chemical compounds across the mycelium. If a nutrient is present at one location and absent at another, a concentration gradient forms. The mycelium can detect this gradient and respond, growing toward the source of the chemical signal. This chemical sensing involves receptor proteins on the surface of fungal cells. These receptor proteins are exquisitely sensitive, capable of detecting extremely low concentrations of specific compounds. When a receptor protein binds to its target molecule, a signal is generated within the cell. This signal is amplified and transmitted throughout the mycelium, allowing the entire organism to respond to a chemical cue detected by a single hypha. The mycelium can distinguish between different nutrients. Not all nutrients are equally valuable. Some provide energy, others provide building blocks for growth, others provide elements like nitrogen or phosphorus that are essential for life, but more difficult to obtain. The mycelium has evolved the ability to detect different nutrients and respond appropriately. If nitrogen is detected, the fungus may respond by increasing its growth rate. If phosphorus is scarce, the fungus may respond by producing compounds that help extract phosphorus from the soil. The mycelium can detect the presence of other organisms. The mycelium is not alone in the soil. It shares its environment with countless other organisms, some potential competitors, some potential partners. The mycelium can detect the presence of other fungi, of bacteria, of plant roots, and respond appropriately. The presence of a competing fungus might trigger the production of antimicrobial compounds. The presence of a plant root might trigger changes in hyphal morphology that facilitate mycorrhizal colonization. Water availability is sensed and responded to by the mycelium. Fungi need water to grow and function. In dry conditions, growth slows or stops. In wet conditions, growth accelerates. The mycelium can sense the water potential of its environment and adjust its physiology and growth accordingly. Some fungi can even produce specialized resting structures when conditions become too dry, structures that can survive desiccation and resume growth when water becomes available again. Temperature is another environmental factor that the mycelium senses and responds to. Different fungal species have optimal temperature ranges for growth. Some fungi are adapted to cool environments, others to warm. Within the optimal range, growth accelerates with increasing temperature. Outside the optimal range, growth slows. Some fungi can sense temperature changes and respond accordingly, even anticipating seasonal changes. The mycelium may prepare for winter by accumulating stored nutrients, or prepare for spring by increasing growth rate when temperatures rise. The mycelium can sense light, at least at the surface or near the surface where light penetrates soil. Some fungi have evolved light-sensing mechanisms that allow them to detect whether they are in darkness or light. This information can affect fruiting body production. Many fungi produce fruiting bodies in response to light, using light as a signal that conditions are favorable for spore dispersal. A mushroom fruiting body is often produced at the soil surface or above it, where spores can be released into the air. The fungus uses light detection to determine when it has reached the surface and can begin fruiting. The mycelium responds to mechanical stress through mechanoreceptors, proteins that sense physical forces. When the mycelium encounters an obstacle, when it is compressed or stretched, these mechanical stresses are detected. The mycelium responds by adjusting its growth direction, by modifying its hyphal morphology, or by producing compounds that help it overcome the obstacle. This mechanical sensing and response allows the mycelium to navigate through complex soil environments around obstacles, along paths of least resistance. Osmotic pressure, the balance between water and dissolved substances, is sensed by the mycelium. Fungi maintain their integrity by regulating osmotic pressure, the balance of water and dissolved compounds across the cell membrane. When external osmotic pressure changes, the mycelium can sense this change and respond by adjusting its internal osmotic pressure. This allows fungi to survive in environments with varying water availability and varying concentrations of dissolved salts. The mycelium can sense the presence of pathogens and parasites through pattern recognition. The fungal immune system has evolved to recognize patterns associated with pathogens and parasites. When these patterns are detected, defensive responses are triggered. These defensive responses might include the production of antifungal compounds, the formation of protective barriers, or even the programmed death of infected cells, a sacrifice to prevent the pathogen from spreading throughout the network. Segment 12. Fungal metabolism and energy production. We now explore the biochemistry of fungal life, the metabolic processes that allow fungi to obtain and utilize energy, to grow, and to perform the countless chemical transformations necessary for life. Fungi are heterotrophs, meaning they obtain energy by consuming organic compounds produced by other organisms. Unlike plants, which capture energy from sunlight through photosynthesis, fungi must obtain their energy from chemical compounds. These compounds might come from living organisms, from dead organic matter, from other fungi, or even from compounds produced by bacteria. The ability to break down diverse organic compounds and extract energy from them is a defining characteristic of fungi. The breakdown of organic compounds by fungal enzymes is the foundation of fungal metabolism. Fungi produce enzymes that can break bonds between atoms in organic compounds, releasing energy that the fungus can capture. These enzymes are specific to particular types of bonds. Cellulase enzymes break down cellulose. Protease enzymes break down proteins. Lipase enzymes break down fats. By producing the right combination of enzymes, a fungus can break down almost any organic compound. The products of enzymatic breakdown are taken up by the fungal cells and processed further. Some products are oxidized, their chemical energy extracted. Other products are used as building blocks for growth, for the synthesis of new cells and hyphae. Still others are stored as reserves, accumulated in the form of glycogen or lipids, to be used later when food is scarce. Fungi can exist in aerobic or anaerobic conditions, though most prefer aerobic conditions where oxygen is available. When oxygen is present, fungi can extract more energy from organic compounds through aerobic respiration. When oxygen is unavailable, some fungi can switch to anaerobic respiration or fermentation, extracting less energy, but still capable of functioning. This metabolic flexibility allows fungi to survive in diverse environments. The efficiency of fungal metabolism varies depending on the substrate being utilized. Some substrates are energetically rich and can be quickly broken down. Others are difficult to degrade and require more enzyme production and more time. A fungus growing on sugar can grow rapidly because sugar is easily metabolized. A fungus growing on cellulose or chitin might grow more slowly because these compounds are resistant to degradation. Yet over time, the fungus can completely break down these difficult substrates, releasing the energy and chemical building blocks locked within them. Fungal growth follows a pattern of phases. Initially, when a fungus colonizes a new substrate, it must synthesize enzymes and other proteins necessary for utilizing that substrate. This lag phase involves metabolic work without much visible growth. Once the enzymatic machinery is in place and the substrate is being actively broken down, the fungus enters an exponential growth phase, where growth is rapid and consistent. Finally, as the substrate becomes depleted or as toxic metabolic byproducts accumulate, growth slows and the fungus enters a stationary phase. During the growth phases, the fungus synthesizes new cell wall material, new membranes, new proteins, nucleic acids, and all the other compounds that make up a living cell. This synthesis requires raw materials obtained from the substrate and energy obtained from the breakdown of the substrate. The mycelium is a factory, continuously processing raw materials and producing new fungal cells and hyphae. The mycelium accumulates secondary metabolites, compounds that are not directly used for growth but provide advantages in the fungus's environment. These include toxins that inhibit competitors, compounds that protect against UV radiation or oxidative stress, compounds that serve as signals in fungal communication, or compounds that have antimicrobial properties. The production of secondary metabolites is energetically costly, yet fungi invest in their production because the benefits outweigh the costs. Nutrient acquisition is a central focus of fungal metabolism. Fungi need nitrogen, phosphorus, potassium, and many other elements to grow. The availability of these nutrients in the environment is highly variable. A fungus might find itself in nutrient-rich soil, but more often it confronts nutrient-poor substrates. Through evolution, fungi have developed sophisticated systems for nutrient acquisition. They produce enzymes that can break down resistant compounds and release bound nutrients. They form partnerships with other organisms that provide access to nutrients they cannot obtain alone. They transport nutrients efficiently throughout their mycelial networks, stockpiling them in nutrient-rich areas and drawing from these reserves in nutrient-poor areas. Segment 13. Fungal genetics and reproduction strategies. We now turn our attention to the genetic basis of fungal life, to the DNA and genes that encode fungal proteins and characteristics, and to the reproductive strategies that fungi employ. Fungi typically have haploid cells, cells with a single copy of each chromosome. This is different from animals and plants, which are typically diploid, with 2 copies of each chromosome. The haploid condition of fungi has interesting implications. It means that mutations are immediately expressed, not masked by a second copy of the gene. This makes fungi sensitive to mutations, but it also means that beneficial mutations can be rapidly selected for and become fixed in the population. Sexual reproduction in fungi involves the fusion of compatible cells, as we have discussed earlier. 2 hyphae of compatible mating types fuse, their genetic material merges, and a diploid cell is briefly formed. This diploid cell can undergo meiosis, a process that shuffles genes and produces spores. The spores are haploid and genetically diverse, each one a unique combination of the genetic material from its 2 parents. When these spores germinate, they produce new haploid fungal organisms with genetic variation that increases the chances that some will survive in changing environments. Asexual reproduction in fungi produces identical copies of the parent fungus. Mycelium can produce spores asexually, without the involvement of sexual processes or genetic mixing. These asexual spores, called conidia, are genetically identical to the parent. Asexual reproduction allows fungi to rapidly colonize new territory, to produce large numbers of spores quickly, to exploit resources before competitors arrive. However, asexual reproduction produces no genetic variation, which can be a disadvantage if the environment changes. Many fungi have both sexual and asexual reproductive strategies, using each in different circumstances. Under favorable conditions, asexual reproduction rapidly produces numerous spores. When conditions become challenging, sexual reproduction and the genetic recombination it entails may be favored, producing genetic diversity that increases the chances that at least some offspring will be suited to the changed conditions. The genome of fungi is typically smaller than the genomes of animals and plants, yet it contains all the genetic information necessary for fungal life. Fungal genomes typically contain several thousand genes. These genes encode the proteins that make up the fungal cell, the enzymes that break down organic compounds, the proteins that allow the fungus to sense its environment and respond to it, and the proteins that carry out all the other functions necessary for life. Gene expression in fungi is dynamic and responsive. A fungus in contact with a plant root will express different genes than a fungus in contact with a competing fungus. A fungus growing on a protein-rich substrate will express different genes than a fungus growing on a carbohydrate-rich substrate. This dynamic gene expression allows fungi to adjust their protein production to their circumstances, to emphasize different functions depending on what is needed. The genetic basis of fungal enzyme production is particularly important. Fungi have genes that encode the enzymes necessary to break down particular substrates. These genes are often organized in clusters and controlled together. When the substrate becomes available, the genes are activated and the enzymes are produced. When the substrate is depleted, the genes are turned off and enzyme production ceases. This regulation prevents the fungus from wasting energy producing enzymes when the substrate is not available. Horizontal gene transfer, which we discussed earlier, has played an important role in fungal evolution and adaptation. Fungi have acquired genes from bacteria, from other fungi, and possibly from other sources. These acquired genes have given fungi new capabilities, allowing them to exploit new substrates, resist antimicrobial compounds, or adapt to new environments. The ease with which fungi acquire new genes, through horizontal gene transfer and through other means, has allowed them to evolve rapidly and adapt to new challenges. Mutation and natural selection continue to shape fungal populations. Mutations are changes in the DNA sequence that can produce new variants of genes and new proteins. Most mutations are harmful or neutral, but some provide advantages. Fungi with advantageous mutations are more likely to survive and reproduce, passing these advantageous mutations to their offspring. Over time, beneficial mutations become more common in fungal populations. This process of natural selection has produced the remarkable diversity and adaptability of fungi we see today. Segment 14. The hidden benefits fungi provide to humanity. Having explored the fungal world in all its complexity and beauty, we now consider the practical and profound benefits that fungi provide to humanity. These benefits are often invisible, hidden from our awareness, yet essential for human survival and well-being. Fungi are essential for agriculture and food production. Without fungal partners, the productivity of agricultural soils would be greatly reduced. Most crops form mycorrhizal relationships with fungi. These relationships improve the plant's ability to obtain water and nutrients, leading to better crop yield and quality. Farmers who understand the importance of fungal partnerships can manage their soils to promote beneficial fungal growth, improving crop productivity without increasing chemical inputs. Fungi are primary sources of food for humans. Many of us have eaten mushrooms, enjoyed their culinary qualities, but fungi provide far more than just the visible mushrooms. Yeasts, which are single-celled fungi, are used to produce bread, beer, wine, and many other fermented foods. These foods are staples in many human diets, and without yeasts, our food culture would be unimaginably different. Fungi produce medicines that have saved countless human lives. Antibiotics like penicillin were discovered in fungi. Since the discovery of penicillin, many other antibiotics have been derived from fungi. These medicines have transformed medicine, allowing the treatment of bacterial infections that once would have been fatal. Fungi continue to be a source of new medicines, with researchers searching fungal genomes and fungal metabolites for compounds that might treat cancer, inflammation, and other human diseases. Fungi are used in industrial processes, breaking down organic materials and producing useful compounds. In textile manufacturing, fungi are used to improve fabric quality. In biofuel production, fungi are used to break down cellulose and convert it to fermentable sugars. In waste treatment, fungi are used to break down pollutants and transform waste into harmless or useful products. The enzymatic capabilities of fungi make them valuable tools in many industrial contexts. Fungi are important indicators of environmental health. The diversity and abundance of fungi in soil is an indicator of soil health. Healthy soils, rich in organic matter and biological activity, support diverse fungal communities. Soils that have been chemically treated, compacted, or otherwise damaged have reduced fungal diversity. By monitoring fungal communities, we can assess the health of soils and ecosystems and identify environmental problems that need to be addressed. Fungi help remediate contaminated environments. Some fungi can break down pollutants, including petroleum products, heavy metals, and other contaminants. Through a process called mycoremediation, fungi can be used to clean up contaminated sites, breaking down pollutants and rendering contaminated soil safer. This is a slow process, but it is often far cheaper and less environmentally disruptive than other remediation methods. Fungi contribute to ecosystem resilience and biodiversity. Mycorrhizal networks, by connecting plants, allow nutrient and information exchange that strengthens plant communities and increases biodiversity. Forest ecosystems with high fungal diversity are often more resilient to disturbances, more productive, and more biodiverse than forests with low fungal diversity. Fungi provide aesthetic and spiritual value to human cultures. For thousands of years, fungi have featured in human art, literature, and spirituality. Mushroom hunting is a traditional practice in many cultures, connecting people to nature and providing food and recreation. The mysterious nature of fungi, their hidden growth, their sudden appearance of fruiting bodies, has inspired human imagination and wonder. Fungi are increasingly recognized as important for human health. The human microbiome includes fungi, and these fungal communities may play roles in human health and disease. Emerging research suggests that imbalances in fungal communities might contribute to allergies, asthma, and other conditions. Understanding and managing fungal communities in the human body might lead to new treatments and improved health. Fungi inspire biotechnological innovations. Researchers are studying fungal networks, fungal enzymes, fungal communication systems, and applying this knowledge to develop new technologies. Mycelium-based materials are being developed as replacements for plastics and leather. Fungal networks are being studied to inspire new communication technologies. The fungal world continues to inspire human innovation and creativity. Segment 15, The Future of Fungal Science and Conservation. As we approach the end of our exploration of the hidden lives of fungi, let us consider what the future holds for fungal science, for our understanding of fungi, and for the conservation and sustainable use of fungal resources. Fungal genomics is rapidly advancing, with the complete genomes of many fungal species now sequenced and available for study. This genomic information reveals the genetic basis of fungal capabilities and adaptations. As more fungal genomes are sequenced, we gain deeper understanding of fungal diversity and evolution. Genomic analysis allows researchers to identify genes responsible for enzyme production, for pathogenicity, for symbiotic relationships, and for other important fungal characteristics. This knowledge opens new possibilities for biotechnology and medicine. The study of fungal communication and coordination within mycelial networks is an emerging field of research. Scientists are investigating how information flows through mycelial networks, how mycelia coordinate activity across vast distances, whether fungi have forms of cognition or intelligence, These studies may reveal new principles of biological organization and may inspire technological innovations in communication and distributed computing. Conservation of fungal diversity is becoming increasingly important. Many fungal species are threatened by habitat loss, climate change, and other environmental factors. Some medicinal fungi that have been harvested for centuries are becoming rare. Conservation efforts are beginning to recognize the importance of protecting fungal diversity, both for its intrinsic value and for the practical benefits that fungi provide to ecosystems and to humans. The development of mycelium-based materials is creating new possibilities for sustainable manufacturing. Mycelium can be grown on agricultural waste, forming a mycelium leather-like material that is biodegradable and has properties similar to leather or plastic. Mycelium-based foam and insulation materials are being developed as sustainable alternatives to petroleum-based products. These developments could reduce human dependence on fossil fuels and non-renewable materials. Mycological restoration, the restoration of fungal communities in degraded ecosystems, is an emerging practice. Scientists and land managers are recognizing that restoring fungal diversity is essential for ecosystem restoration. By inoculating degraded soils with beneficial fungi, by creating conditions that favor fungal colonization, restoration efforts can rebuild the fungal networks that form the foundation of healthy ecosystems. The role of fungi in climate regulation and carbon cycling is being increasingly appreciated. Fungi play important roles in storing carbon in soils, in regulating nutrient cycling that affects plant productivity and carbon sequestration. As concerns about climate change grow, the role of fungi in climate regulation is receiving increased attention. Understanding and managing fungal communities might contribute to climate change mitigation. Fungal-based agriculture, agriculture that emphasizes the role of beneficial fungi, is being developed and promoted. Farmers and gardeners are learning how to cultivate beneficial fungal relationships to reduce dependence on chemical inputs while maintaining or increasing productivity. As chemical agriculture becomes increasingly questioned for its environmental impacts, fungal-based agriculture offers a sustainable alternative. The study of human fungal microbiomes is revealing the importance of fungi for human health and well-being. The fungi that live on our skin, in our respiratory systems, in our digestive systems, are not merely passengers, but active contributors to human health. Understanding these fungal communities and how to maintain healthy fungal communities might lead to new treatments for diseases and new approaches to promoting human health. Citizen science initiatives are engaging the public in fungal research. Mushroom foragers, nature enthusiasts, and amateur naturalists are contributing valuable data about fungal distributions, fruiting patterns, and ecological relationships. These citizen science initiatives democratize fungal research and raise awareness about the importance of fungi. The integration of fungal knowledge into education at all levels is important for building understanding and appreciation of fungi. As fungi are often neglected in science education, efforts to increase fungal literacy are underway. Educational programs, nature guides, and popular science books about fungi are helping people understand the hidden lives of fungi and their importance for life on Earth. Finally, we recognize that there is still so much we do not know about fungi. The vast majority of fungal species have not yet been described by science. The ecological roles of many fungi are poorly understood. The full extent of fungal capabilities and intelligence is only beginning to be appreciated. The fungal world remains largely hidden, and new discoveries are constantly being made. The future of fungal science is bright with possibility, with new questions to investigate, new phenomena to understand, new ways to appreciate and utilize the remarkable kingdom of fungi. Segment 16: The Soil Beneath Our Feet. Let us now turn our attention to the soil itself, that rich, dark medium in which so much of fungal life occurs. Soil is not merely inert matter, not just broken down rock and dead organic material. Rather, soil is a living system, and fungi are central to making soil the living, fertile substrate that supports terrestrial life. The formation of soil takes thousands of years, and fungi play a central role in this long, slow process. When rock is exposed to the weathering elements, water, temperature fluctuations, and chemicals slowly break it down into smaller particles. But this physical and chemical weathering alone is not enough to create fertile soil. Life must be involved. Fungi are among the first colonizers of barren rock. Their acids, their enzymes, their biological activity begin the transformation of bare mineral into living soil. As fungi break down the minimal organic matter available and gradually accumulate it in the rock's crevices, they create conditions where other organisms can establish themselves. Over time, a succession of organisms colonizes this developing soil. Bacteria arrive, cyanobacteria begin fixing atmospheric nitrogen, lichens composed of fungi and algae establish themselves. Plants gradually become able to grow, and their roots penetrate the developing soil, adding organic matter when leaves and roots die. Through all of this succession, fungi remain central, breaking down dead material, creating soil structure, facilitating nutrient cycling. A thin layer of living soil gradually develops, and it is fungal networks that hold this soil together that make it capable of supporting plant growth. The soil food web is a complex network of organisms with fungi at or near its center. At the base of this food web are photosynthetic organisms and organic matter from dead organisms. Fungi feed on this organic matter, breaking it down and obtaining energy. As fungi break down organic matter, they produce byproducts and their own biomass, which feed other organisms. Bacteria consume some of the organic matter and also feed on fungal byproducts. Protozoa consume bacteria, nematodes consume fungi and bacteria, arthropods consume fungi and other soil organisms. Larger animals feed on soil invertebrates. At each step, energy flows from one level to the next, but much energy is also lost as heat. This complex food web, with its multiple paths and connections, is what makes soil ecosystems resilient and productive. Fungal decomposition in soil is not a simple linear process. Rather, different fungi with different capabilities become active at different stages of decomposition. Fresh plant material is initially colonized by pioneer fungi that can tolerate the acidic and antimicrobial compounds in fresh plant tissue. As these pioneer fungi break down the easier-to-access compounds, later arriving fungi specialize in breaking down more resistant compounds like cellulose and lignin. Eventually, the most resistant compounds are broken down by the late-stage decomposers, and what remains is well-processed, incorporated organic matter thoroughly mixed with mineral soil. The mycelial networks in soil create aggregates, clumps of soil particles bound together by fungal hyphae and fungal exudates. These soil aggregates have profound importance for soil function. Aggregates increase soil porosity, creating spaces where water can move and where air can penetrate. Aggregates increase soil water holding capacity, allowing soil to retain moisture that plant roots can access during dry periods. Aggregates reduce soil erosion as water moves through the aggregated soil more gradually than through non-aggregated soil. Soils with high fungal activity and abundant mycelial networks tend to have excellent soil structure, excellent aggregation, and excellent properties for plant growth. Fungal decomposition also contributes to the development of soil profile, the layering of soil that develops over time. As organic matter is incorporated into the upper layer of soil, this layer becomes enriched with organic matter and becomes darker in color. Below this is a layer where minerals accumulate as they are leached from the upper layer. Below that is the parent material, the partially weathered rock from which the soil originated. As soil develops, these layers become more distinct, more differentiated, reflecting the different processes occurring in different parts of the developing soil profile. The texture of soil, the proportion of sand, silt, and clay particles, is partly determined by fungal activity. Fungi don't directly change soil texture by breaking down sand and silt and clay particles, but they do affect how these particles interact. Fungal exudates coat soil particles, causing them to stick together. This effectively increases the apparent size of particles, making a sandy soil more cohesive, more able to retain water. In this way, fungal activity influences the physical properties of soil beyond just the organic matter content. The color of soil is often an indicator of fungal and biological activity. Dark soils, rich brown or black in color, indicate high organic matter content and high biological activity, usually including extensive fungal presence. Light-colored soils, by contrast, often indicate lower organic matter and lower biological activity, including less fungal presence. The color of soil is thus a window into the living processes occurring within it, a visible reminder that soil is a living system, not merely inert material. Segment 17, Fungal Diversity Across Ecosystems. Having explored fungal roles and processes, let us now consider the extraordinary diversity of fungi across different ecosystems and environments. The fungal kingdom is so vast and varied that no single chapter, no single video could possibly encompass the full diversity of fungal life on Earth. Temperate forests are among the richest ecosystems in terms of fungal diversity. The cool, moist conditions of temperate forests, combined with abundant plant diversity, create ideal conditions for fungal growth. In a temperate forest, hundreds or thousands of fungal species might be present in a single hectare of land. Many of these fungi form mycorrhizal relationships with trees. Others are saprotrophs, decomposing dead wood and leaf litter. Others are parasites or pathogens, feeding on living organisms. Still others are insect associates, growing on insect bodies or in insect nests. The temperate forest is a mycological paradise, a place where fungi are abundant and diverse. Tropical rainforests, despite their abundance of plant life and biodiversity, often present challenges for fungal growth. The warm, moist conditions favor fungal growth, but they also favor the growth of competitors. Bacteria are more aggressive in tropical conditions, and bacterial decomposition occurs more rapidly than in cooler climates. Yet despite these challenges, tropical fungal diversity is extraordinary. Tropical fungi have evolved specialized forms, specialized niches, ways of competing in a highly competitive environment. Many tropical fungi produce fruiting bodies with remarkable colors and forms, adaptations that make sense in the context of insect dispersal in the complex tropical forest environment. Grasslands and savannas present different challenges for fungi. The drier conditions, the dominance of grasses, the higher temperatures, at least in savannas, all these create different selective pressures than forest environments. Yet grassland fungi have adapted, often forming extensive mycorrhizal relationships with grasses, sometimes forming arbuscular mycorrhizal associations rather than the ectomycorrhizal associations common in forests. Grassland fungi include species specialized for breaking down grass roots, for living in dry soil, for surviving periods of drought. Desert fungi are extreme specialists, adapted to survive in some of the harshest conditions on Earth. Yet deserts are not fungal deserts. Rather, deserts support fungal communities that have evolved remarkable adaptations to aridity. Some desert fungi form lichens with photosynthetic partners, creating self-sufficient organisms that can grow on bare rock. Some desert fungi form associations with desert plants, helping them access the limited water and nutrients available. Some desert fungi produce resistant spores that can remain dormant for years, waiting for the rare rainfall events that make growth possible. Arctic and alpine environments present challenges of cold and a short growing season. Yet fungi thrive in these extreme environments, adapted to work within the short window of warmth and light that these regions experience. Some Arctic fungi form lichens that are among the few visible plants in Arctic tundra. Others form symbiotic relationships with Arctic plants, enabling their survival in nutrient-poor soils. Some Arctic fungi produce antifreeze proteins that keep their cells from freezing, even at temperatures well below zero. Aquatic fungi, both in freshwater and marine environments, represent an entirely different branch of fungal diversity. Aquatic fungi have evolved mechanisms to deal with the challenges of living in water, including osmotic regulation and adaptations to high water movement. Some aquatic fungi are aquatic in all their life stages. Others are primarily terrestrial, but have adapted to occasional water exposure. Aquatic fungi play important roles in decomposition and nutrient cycling in aquatic ecosystems, though they receive far less attention than terrestrial fungi. Fungi associated with deadwood represent a specialized and important group. Different fungal species colonize wood at different stages of decomposition. Pioneer colonizers establish themselves in recently dead wood. Later colonizers break down the tougher compounds like lignin. Final colonizers completely break down the remaining material, converting dead wood into soil. Without wood-decomposing fungi, dead trees would accumulate. Forests would be clogged with dead timber. Nutrient cycling would be severely impaired. Cave fungi represent another specialized fungal ecology. Caves are dark, cool, isolated environments. Fungi in caves have adapted to extremely low light conditions and often depend on nutrient input from outside the cave system. Cave fungi are often colorless or lightly pigmented, having no need for pigments that protect against light damage when light never reaches them. Cave fungi help break down bat guano and other organic matter that enters caves, contributing to nutrient cycling in these isolated ecosystems. Segment 18: The Mycelial Networks of Ancient Forests. Let us take a moment to consider in detail the fungal ecology of ancient forests, where fungi have had thousands of years to establish their networks and relationships. These ancient forests are places where fungal networks reach their most developed and complex forms. In old-growth forests, fungal diversity reaches its peak. These forests contain fungi that have been establishing relationships with trees for hundreds of years. The mycorrhizal networks in these forests are ancient, extensive, deeply integrated with the forest ecosystem. Trees in old-growth forests, particularly late successional species that establish themselves only after pioneer species have modified the environment, are completely dependent on these established mycorrhizal networks. The relationship between these ancient trees and their fungal partners is so intimate, so essential, that the trees and fungi might be considered as a unified system rather than as separate organisms. The mycelial networks in ancient forests facilitate nutrient cycling in ways that are far more efficient than in younger forests. Young forests, with their simple structure and fewer fungal species, cycle nutrients relatively slowly. Ancient forests, with their complex fungal networks, their numerous fungal species, each specialized for different ecological roles, cycle nutrients far more efficiently. Nutrients from dead leaves and wood are rapidly broken down and made available to living trees. Nutrients captured by fallen leaves are efficiently transferred through mycorrhizal networks to living trees that need them. In ancient forests, we often find the fruiting bodies that result from thousands of years of mycelial network establishment. Large, old mushroom fruiting bodies, massive woody shelf fungi growing from tree trunks, distinctive mushrooms that fruit year after year in the same locations, all these are visible manifestations of the ancient fungal networks that exist beneath the forest floor and within the wood of trees. These fruiting bodies are old in the sense that the mycelial networks that produce them may be centuries or millennia old, though the fruiting body itself is produced anew each year. The resilience of ancient forests is substantially dependent on fungal networks. When an old tree falls in an ancient forest, the mycelial networks associated with that tree help ensure that its nutrients are recycled back into the forest, available to other trees and plants. The mycelial networks in healthy ancient forests are so extensive that they enable the forest to recover from disturbances that would devastate younger forests. A young forest might be set back decades or centuries by a logging operation or a major wildfire. An ancient forest, with its extensive fungal networks and its numerous tree species, can recover far more quickly because the fungal infrastructure remains intact and is ready to support the regeneration of forest vegetation. Ancient forests also contain fungi that cannot survive in young, recently disturbed forests. Some fungi require specific conditions that exist only in undisturbed, old-growth forest. Some fungi have specialized relationships with old-growth tree species. Some fungi are so slow-growing that they require centuries to establish themselves. When old-growth forests are destroyed, they lose not only trees, but fungal species that may never be replaced. The fungal diversity of ancient forests is irreplaceable in any meaningful human timescale. The study of ancient forests is revealing the incredible complexity of fungal networks in these systems. Using molecular techniques, scientists can now map mycelial networks and identify the fungal species present. What they find is remarkable. A single cubic centimeter of soil in an ancient forest may contain mycelia from dozens or hundreds of fungal species. These fungi occupy different ecological roles, use different substrates, associate with different plant partners. This extraordinary diversity is one of the hallmarks of ancient forest fungal ecology. Segment 19: Mycorrhizal Networks as Information Highways. We have discussed mycorrhizal networks as a vehicle for nutrient transport, but there is growing evidence that these networks also function as information highways, allowing plants to communicate with each other and with their fungal partners in ways we are only beginning to understand. The chemical signals that flow through mycorrhizal networks carry information about environmental conditions, nutrient availability, and the presence of stresses like herbivory or pathogen attack. When a plant is attacked by insect herbivores, it can produce volatile compounds that signal the presence of the herbivores. Some of these volatiles are released into the air, but others can be transmitted through mycorrhizal networks to neighboring plants, warning them of the herbivore attack so they can mount their own defensive responses. This is a form of plant communication mediated by fungi that allows plants to share information about threats. The mycorrhizal network can also transmit information about nutrient availability. A plant in a nutrient-rich location that is producing excess photosynthetic products can transfer these through the mycorrhizal network to a nutrient-poor location where a partner plant is struggling. In doing so, the abundant plant is signaling to its fungal partner that nutrients are plentiful, and the fungal partner responds by reallocating its resources. The nutrient transfer is driven by the concentration gradients created by photosynthesis and nutrient uptake, but the direction of nutrient flow can be influenced by signaling between partners. Some researchers have speculated that mycorrhizal networks might constitute a form of plant internet, allowing plants to communicate across distances, sharing information and resources. While this poetic terminology might overstate what we currently know, it captures something important about the function of these networks. Plants in a forest are not isolated individuals, but rather members of a community connected through fungal networks, capable of sharing resources and information in ways that benefit the community as a whole. The identity of the plants connected by a mycorrhizal network influences the resources that are transferred. Some plants, particularly fast-growing pioneer plants, are net exporters of photosynthetic products, transferring resources to slower-growing plant partners. Some plants, particularly shade-tolerant species that produce less photosynthetic product, are net importers, receiving resources from more productive partners. Over time, This differential resource allocation can influence the competitive relationships between plants and can shape the composition of plant communities. The mycorrhizal network is not a neutral conduit, but an active participant in resource allocation. The fungal partners in mycorrhizal networks have their own interests and their own strategies for maximizing their own growth and reproduction. The fungus does not simply passively transfer resources from one plant to another. Rather, the fungus makes decisions, in a sense, about how to allocate resources, about which plant partners to prioritize. Some fungi preferentially support the most productive plants, maximizing the resources they themselves receive. Other fungi might support plants that are struggling, ensuring the survival of plant partners they have invested heavily in. Segment 20: The future we are creating. As we contemplate the future of fungi and fungal ecology, we must recognize that fungi face unprecedented challenges and unprecedented opportunities. The choices we make in the coming years and decades will profoundly shape the future of fungal diversity and fungal ecology. Climate change is altering the conditions in which fungi live. Changing temperatures and precipitation patterns are shifting the geographic ranges of fungal species. Some fungal species may expand their range as conditions become more favorable for them. Others may be pushed toward extinction as their optimal conditions disappear. The complex webs of fungal associations with plant and animal partners may unravel as climate change disrupts the synchrony between partners that have co-evolved over long periods. We are likely to see significant reshuffling of fungal communities in response to climate change, with winners and losers among fungal species. Agricultural practices influence fungal diversity profoundly. Industrial agriculture, with its emphasis on monocultures, chemical fertilizers, and pesticides, generally reduces fungal diversity and disrupts mycorrhizal relationships. The short-term productivity gains of industrial agriculture may come at the cost of long-term fungal diversity and soil health. Conversely, agricultural practices that emphasize soil health, that minimize chemical inputs, that promote fungal diversity, may be more sustainable in the long term and better suited to addressing the challenges of the future. Habitat destruction and land use change are causing the loss of fungal diversity at an alarming rate. As forests are cleared, as wetlands are drained, as grasslands are converted to agriculture, the fungal species associated with these ecosystems are being lost. Many fungal species have never been described by science, and they may be going extinct before we even know they existed. The loss of fungal diversity is occurring at a pace far greater than our capacity to document it, let alone to conserve it. Conservation of fungal diversity is increasingly being recognized as a priority. Protected areas, if they include old-growth forests and other diverse ecosystems, can serve as refugia for fungal diversity. Restoration efforts that focus on re-establishing mycorrhizal networks and fungal communities can help restore ecosystem function in degraded areas. Seed banking and genetic preservation of fungal strains can help ensure that fungal diversity is not completely lost, even if wild populations disappear. The potential of fungi to help address human challenges is enormous. Fungi can help remediate contaminated sites, breaking down pollutants, and making degraded environments functional again. Fungi can be used to produce biofuels, breaking down cellulose and converting it to fermentable sugars. Fungi can produce medicines that treat diseases that currently have no good treatments. Fungi can provide sustainable materials, mycelium-based products that replace petroleum-based plastics and other materials. As we face challenges of pollution, energy, disease, and sustainability, fungi offer potential solutions that are grounded in billions of years of evolution. The integration of fungal science into environmental decision-making and policy is essential for a sustainable future. Conservation policies that recognize the importance of fungi, that protect old-growth forests where fungal diversity is greatest, that minimize the disruption of mycorrhizal networks, will lead to better conservation outcomes. Agricultural policies that promote soil health and fungal diversity will lead to more sustainable food production. Industrial policies that recognize the potential of fungi to help solve environmental problems will accelerate innovation and sustainable development. Education about fungi is critical for creating a society that values and protects fungal diversity. Most people know little about fungi beyond the mushrooms they might eat. Increasing fungal literacy, teaching people about the roles fungi play in ecosystems and in human life, can help create public support for fungal conservation and research. Nature guides, documentaries, field courses, and informal education about fungi can help people understand and appreciate the fungal world. Segment 21. Fungal partnerships with specific plant families. To deepen our understanding of mycorrhizal relationships, let us explore the specific partnerships that fungi have developed with different plant families. Different plant groups have evolved different strategies for working with fungi, and these different strategies have profound implications for how these plants survive and thrive in various environments. The orchid family represents perhaps the most specialized mycorrhizal relationships known to science. Orchids are renowned for their extraordinary floral beauty, but many orchid species have equally extraordinary relationships with fungi. Orchid seeds are tiny and contain minimal stored nutrients. For an orchid seed to germinate and survive, it must be colonized by a compatible fungus. The fungal partner provides essential nutrients to the developing seedling, allowing it to grow until it can photosynthesize and support itself. This relationship is so specific and so necessary that removing the fungal partner often kills the orchid plant. In nature, orchid distribution and orchid diversity are limited by the availability of compatible fungal partners. Where suitable fungi are absent, orchid species cannot establish themselves, no matter how suitable the soil or climate might otherwise be. The majority of temperate forest trees, particularly the conifers and hardwoods of the northern hemisphere, form ectomycorrhizal partnerships with fungi. In these partnerships, the fungal hyphae form a sheath around the plant root, rather than penetrating into the root cells as occurs in other mycorrhizal types. This ectomycorrhizal association allows trees to access soil minerals in very nutrient-poor soils, soils where trees without fungal partners would struggle. Many valuable timber trees, including pine, spruce, fir, beech, and oak, depend on ectomycorrhizal fungi. The large, visible mushrooms that appear in forests in autumn, the prized edible mushrooms that many people hunt and consume, are the fruiting bodies of ectomycorrhizal fungi. The relationship between these mushroom-producing fungi and the trees is ancient and essential. Grasses, including the economically important cereal crops like wheat, rice, and corn, typically form arbuscular mycorrhizal associations with fungi. In these partnerships, fungal hyphae actually penetrate into the root cells, forming tiny ball-shaped or branched structures called arbuscules that are the site of nutrient exchange between fungus and plant. This mycorrhizal type is ancient and widespread, occurring in most plant families. The arbuscular mycorrhizal fungi are particularly important for agricultural systems, as these partnerships improve plant nutrient acquisition and plant tolerance of environmental stress. Many agricultural researchers believe that the decline of arbuscular mycorrhizal fungi in industrial agricultural soils is a significant factor in reduced soil health and reduced crop resilience in these systems. The legume family, which includes peas, beans, and other nitrogen-fixing plants, has evolved a different strategy. While legumes often form mycorrhizal relationships with fungi, they more famously form partnerships with nitrogen-fixing bacteria that live in root nodules. These bacteria capture atmospheric nitrogen and make it available to the plant in exchange for carbohydrates. This legume-bacteria partnership allows legumes to grow in nitrogen-poor soils without requiring nitrogen inputs from fertilizers. The combination of bacterial nitrogen fixation and fungal nutrient acquisition makes legumes incredibly productive and resilient plants, which is why they are so widely cultivated and why they feature prominently in sustainable agricultural systems. The mycorrhizal relationships between fungi and plants are not static or unchanging. Rather, they can vary depending on environmental conditions and the specific circumstances of the plant and fungal partners. A plant species might form partnerships with multiple fungal species. The identity of the fungal partner can change over the course of the plant's life, with seedlings forming associations with different fungi than adult plants. When environmental conditions change, the balance between different fungal partners can shift, with some fungal species declining and others becoming more prominent. These specifics of mycorrhizal relationships help us understand the complexity and sophistication of plant-fungal partnerships. They also help us understand why preserving mycorrhizal fungal diversity is so important. If a specific plant species depends on a specific fungal partner, and that fungal partner is lost due to habitat destruction or climate change, the plant species may not be able to survive, even if the plant's other needs are met. Conversely, by understanding these partnerships, scientists can work to preserve both plant and fungal partners together, understanding that the preservation of one requires the preservation of the other. Segment 22: Fungal diseases and ecological balance. While we have focused much of our attention on the beneficial roles that fungi play, we must also acknowledge that some fungi cause disease in plants and animals. These fungal diseases are not aberrations or failures of the fungal kingdom, but rather natural phenomena that play important roles in ecological balance and evolution. Fungal plant diseases range from leaf spots and blights to wilts and root rots. Different fungal pathogens specialize in attacking different plant tissues and different plant species. When a plant is infected by a fungal pathogen, the fungus benefits by obtaining nutrition from the plant tissue. The plant, however, is harmed by the loss of tissue and the disruption of normal physiological processes. Yet in natural ecosystems, fungal plant diseases do not typically kill all individuals of a plant species or drive species to extinction. Rather, fungal diseases exert selective pressure that shapes plant populations and plant communities. Some plants have evolved resistance to fungal diseases through natural selection. Plant varieties that have genetic traits that protect them from fungal attack are more likely to survive and reproduce when fungal diseases are present. Over time, the frequency of disease resistance genes increases in plant populations exposed to fungal diseases. This is an example of coevolution, where the evolution of pathogens drives the evolution of host defenses, which in turn drives the evolution of new pathogen strategies to overcome those defenses. This ongoing arms race between plants and pathogens is one of the driving forces of evolution in natural ecosystems. Fungal diseases of animals and humans are less common than fungal diseases of plants, but they do occur. Fungi can colonize the skin, the respiratory system, and the digestive system of animals. In healthy immune systems, these fungal colonizations are usually kept under control and cause no harm. However, in immunocompromised individuals, or in conditions where fungal growth is particularly vigorous, fungal infections can become problematic. Some fungal diseases in animals can be serious and even life-threatening, though in most cases, fungal infections are benign or cause only minor symptoms. The role of fungal diseases in maintaining ecological balance is important. Fungal diseases can limit the population growth of particular plant or animal species, preventing any one species from completely dominating an ecosystem. Fungal diseases can maintain genetic diversity in plant populations by selecting for disease-resistant genotypes. Fungal diseases can create landscape heterogeneity, with areas of dead or dying plants creating gaps in vegetation that are colonized by other species. In forests, fungal diseases of trees play important ecological roles. Fungi that kill or weaken particular tree species can prevent those species from becoming dominant, allowing other species to persist. Fungal decomposers of dead wood are essential for recycling the nutrients in dead trees. Without fungal decomposers, dead trees would persist indefinitely, clogging forests and tying up nutrients. The presence of fungal diseases and fungal decomposers helps maintain the dynamic balance that characterizes healthy forest ecosystems. That said, invasive fungal pathogens introduced by human activity can cause serious ecological problems. When a fungal pathogen is introduced to a new region where plant species have not evolved resistance to that pathogen, the results can be catastrophic. The fungal pathogen can wipe out entire populations of susceptible plant species, causing massive changes in forest structure and ecosystem function. The American chestnut, once a dominant tree species in eastern North American forests, was nearly driven to extinction by an invasive fungal pathogen introduced from Asia. Similarly, several tree species in Europe and Australia have been threatened by invasive fungal pathogens. These examples illustrate the potential for fungal pathogens to cause ecological damage when they are introduced outside their native range. Segment 23: Fungal Responses to Environmental Stress. We have discussed how fungi sense and respond to their environment, but let us explore more deeply the specific ways that fungi respond to environmental stresses and how these responses help fungi survive in challenging conditions. When mycelium experiences water stress due to drought, it responds through several mechanisms. First, the mycelium can adjust its water potential, the concentration of dissolved substances within its cells, to help retain water. Second, the mycelium can produce specialized structures or alter the composition of its cell walls to reduce water loss. Third, the mycelium can slow its growth rate, reducing its metabolic activity and therefore its need for water. Some fungi can enter dormancy, a state of greatly reduced metabolic activity, allowing them to survive extended droughts without significant damage. When water becomes available again, the dormant mycelium can resume growth and normal activity. Osmotic stress, caused by high concentrations of salts or sugars in the environment, is another challenge that fungi must contend with. Fungi respond to osmotic stress by accumulating compatible solutes, compounds that help maintain cellular water balance without disrupting normal cellular functions. These compatible solutes might include glycerol, amino acids, or sugars. By accumulating compatible solutes, fungi can maintain their internal water potential even in environments with high external osmotic pressure, allowing them to survive in salt-rich soils, salt marshes, or other hypersaline environments. Oxidative stress, caused by the accumulation of reactive oxygen species, is a challenge fungi face when exposed to UV radiation, high temperatures, or other stresses that disrupt normal metabolism. Fungi respond to oxidative stress by producing antioxidant compounds and enzymes that break down reactive oxygen species before they can damage cellular components. Some fungi also produce pigments, such as melanin, that protect against UV radiation and that also help the fungus cope with oxidative stress. Temperature stress, both from cold and heat, is another environmental challenge. Different fungal species have different temperature tolerances and temperature optima. Some fungal species are psychrophilic, adapted to cold environments and growing optimally at low temperatures. Other fungi are thermophilic, adapted to hot environments and capable of growing at temperatures that would kill most other organisms. Most fungi fall somewhere in the middle, with temperature optima in the range of 15 to 30 degrees Celsius. When temperatures exceed the fungus's tolerance range, growth ceases, and the fungus may enter dormancy or may produce resistant spores that can survive the temperature stress. Chemical stress, caused by the presence of compounds toxic to the fungus, is yet another challenge. These toxic compounds might be produced by competing organisms, might be pollutants, might be antimicrobial compounds produced by plants or by the fungus itself. Fungi respond to chemical stress through several mechanisms. Some fungi can detoxify harmful compounds, breaking them down or sequestering them so they cause no damage. Some fungi can upregulate the production of cell wall components that protect against chemical entry. Some fungi can alter their metabolism to bypass the pathways affected by the toxic compound. Through these various mechanisms, fungi can survive in environments containing compounds that would kill most other organisms. Segment 24. The fungal immune system. Just as animals and plants have immune systems, fungi also possess sophisticated mechanisms for detecting and responding to threats from pathogens and parasites. These fungal immune mechanisms are less well understood than immune systems in animals and plants, but they are no less important for fungal survival. Pattern recognition is the first line of fungal defense. Fungi have evolved receptors that recognize molecular patterns associated with pathogens. These patterns might include particular proteins found in fungal competitors or in parasitic organisms. When these patterns are detected, the fungus mounts an immune response. This pattern recognition system allows the fungus to distinguish between friend and foe, to recognize potential threats and initiate defensive responses. The production of secondary metabolites is an important part of fungal immunity. Many of the compounds that fungi produce as secondary metabolites, compounds not directly involved in growth or reproduction, have antimicrobial properties. These compounds can inhibit the growth of competing fungi, can kill bacteria, can deter grazing by animals. The production of these antimicrobial compounds is part of the fungus's immune defense, a chemical arsenal deployed against threats to the fungus's survival. Programmed cell death, or apoptosis, is a dramatic but important fungal immune response. When a fungus is infected with a pathogen, the infected hyphal cells can undergo programmed death, sacrificing themselves to prevent the pathogen from spreading throughout the mycelial network. This is an extreme response, but it can prevent the loss of the entire fungal organism. By killing itself, an infected cell prevents the pathogen from using that cell to spread to other parts of the network. The cell wall itself plays an important role in fungal immunity. The fungal cell wall, composed of chitin and beta-glucans, provides a physical barrier against pathogens and parasites. Some pathogens can break through this barrier, but the cell wall provides important protection. Additionally, the components of the cell wall, particularly beta-glucans, can activate the fungus's immune system when they are damaged, or when pathogenic organisms try to degrade the cell wall. In this way, damage to the cell wall signals the presence of a threat, and the fungus mounts an appropriate immune response. The interconnected nature of mycelial networks allows for rapid distribution of immune signals throughout the organism. When one hyphal cell detects a pathogen or stress, it can produce chemical signals that spread throughout the mycelial network, alerting the entire organism to the threat. This allows the fungus to mount a coordinated immune response across its entire body, even though the network may span vast distances. Some fungi have evolved specific immune responses to particular pathogens that they commonly encounter. Just as animals produce antibodies specific to particular pathogens, fungi can upregulate the production of specific antimicrobial compounds in response to encountering particular pathogenic threats. This specificity allows fungi to mount an immune response that is precisely targeted at the threat, rather than mounting a general immune response that affects all aspects of fungal metabolism. Segment 25: The Future of Human Fungal Collaboration. As we contemplate humanity's future, we must recognize that fungi are not obstacles to be overcome or conquered, but rather partners to be understood and worked with. The future of sustainable human civilization may well depend on our ability to collaborate with and leverage the remarkable capabilities of fungi. Fungal biotechnology is one of the most promising areas of human-fungal collaboration. Scientists are learning to harness fungal enzymes and fungal metabolic pathways for industrial applications. Cellulose enzymes from fungi are being used to break down plant cellulose in biofuel production, potentially offering a sustainable alternative to fossil fuels. Fungal enzymes are being used to produce fine chemicals and pharmaceuticals, reducing the need for chemical synthesis. Fungi are being used to produce plant-based proteins and other meat alternatives, helping to feed a growing human population with reduced environmental impact. As biotechnology advances, the applications of fungal biology are likely to expand dramatically. Fungal agriculture represents another promising area of collaboration. Agricultural systems that work with fungi rather than against them, that deliberately promote beneficial fungal communities in soil, that use mycorrhizal fungi to improve nutrient acquisition by crops, are being developed and implemented. These systems show promise for increasing agricultural productivity while reducing the need for chemical inputs, making agriculture more sustainable and more resilient to environmental challenges. As our understanding of fungal plant partnerships deepens, the integration of fungi into agricultural systems is likely to become increasingly sophisticated and increasingly widespread. Fungal materials science is an emerging field with remarkable potential. Mycelium can be grown on agricultural waste, forming a material with properties similar to leather or plastic, but fully biodegradable. This mycelium material can be used to create clothing, furniture, packing materials, and countless other applications. Unlike petroleum-based plastics, mycelium-based materials can be composted at the end of their useful life, returning their nutrients to soil. As mycelium-based materials are developed and refined, they may eventually replace many petroleum-based products, offering a sustainable alternative that is produced through the growth of a living organism rather than through industrial chemical synthesis. Fungal remediation offers solutions to environmental pollution challenges. Fungi can break down petroleum products, absorbing and degrading crude oil and its derivatives. Fungi can accumulate heavy metals from contaminated soil and water, concentrating them in fungal tissue that can then be harvested and the metals recovered. Fungi can degrade persistent organic pollutants, compounds that resist bacterial decomposition. Using fungi to remediate contaminated sites, a process called mycoremediation, is often cheaper and less environmentally disruptive than traditional remediation methods. As our understanding of fungal decomposition capabilities improves, mycoremediation will likely become an increasingly important tool in environmental restoration. Fungal medicine is likely to produce new therapeutics. Fungi have already provided us with antibiotics like penicillin, saving countless lives. But fungal genetic diversity has barely been scratched. Thousands of fungal species have never been studied for bioactive compounds. As researchers conduct drug discovery studies on fungal species, new medicines for treating cancer, inflammation, infectious diseases, and other conditions may be discovered. Some researchers are exploring whether compounds produced by certain fungi might help treat neurodegenerative diseases or psychiatric conditions. The therapeutic potential of fungi is enormous and largely untapped. Fungal education and public understanding are critical for realizing these benefits. Most people still think of fungi primarily as mushrooms, and many don't realize the central importance of fungi in ecosystems and in human life. By increasing public understanding of fungi through nature guides, documentaries, social media, and other educational media, we can create public support for fungal research and fungal conservation. We can help people understand that protecting fungal diversity is protecting the foundation of terrestrial life. The integration of traditional knowledge about fungi with modern scientific understanding offers another avenue for collaboration. In many cultures, fungi have been used for food and medicine for centuries, or even millennia. Indigenous peoples have knowledge about edible, medicinal, and toxic fungi that has been accumulated over generations. Modern science is beginning to validate some of this traditional knowledge and is learning from indigenous peoples about how to identify and use fungi. By combining traditional knowledge with modern scientific methods, we can accelerate the discovery of new applications of fungi and can help ensure that traditional knowledge is preserved and honored. Government policy and regulation will play important roles in enabling human-fungal collaboration. Environmental policies that protect old-growth forests and other fungal-rich ecosystems will help preserve fungal diversity. Agricultural policies that encourage farming practices that promote soil fungal communities will help maintain soil health. Intellectual property policies that allow researchers and companies to develop and commercialize fungal-based products will accelerate innovation. Investment in fungal research, both basic science and applied research, will accelerate the discovery of new fungal applications. Reflections on the invisible majority. As we move toward the conclusion of our exploration, let us pause and reflect on one of the most striking facts about fungi. They are invisible. The vast majority of fungal life, the mycelial networks that form the foundation of terrestrial ecosystems, are completely invisible to the naked eye. We walk on mycelium, we breathe in fungal spores, we interact with fungi in countless ways, yet most of the time we are unaware of their presence. This invisibility has consequences for human awareness and human action. We tend to care about and protect things that we can see, that capture our attention and imagination. The large animals, the colorful plants, the dramatic geological features, these capture human attention and shape human values. By contrast, the invisible mycelial networks that form the foundation of ecosystem function tend to be overlooked, undervalued, and underprotected. It is easier to care about saving a charismatic megafauna, a symbol of wilderness and natural wonder, than it is to care about protecting fungal networks that we cannot see and that seem abstract and foreign. Yet if we understand the roles that fungi play, if we recognize the dependence of all terrestrial life on fungal networks, we might begin to shift our priorities. We might come to see the protection of fungal diversity as equally important as the protection of visible wildlife. We might recognize that a hectare of old-growth forest, with its ancient and diverse fungal networks, is worth preserving not only for its trees and visible plants, but for its fungal diversity and the ecosystem functions that diverse fungal networks provide. The invisibility of fungi has also shaped the history of scientific understanding. For a long time, fungi were poorly studied and poorly understood. Mycology, the study of fungi, is a younger science than zoology or botany. Fungi lacked the charisma of animals or the obvious importance of plants in producing food and oxygen. It is only in recent decades that the central importance of fungi in ecosystem functioning has become widely recognized in scientific circles. We are living through a revolution in our understanding of fungi, a time when knowledge is accumulating rapidly, and our understanding is being fundamentally reshaped. The future will likely bring increased recognition of the importance of fungi and increased investment in fungal research and conservation. As we face environmental challenges, as we seek sustainable solutions to problems of pollution, energy, and food production, fungi will increasingly be seen as important resources and important partners. The microscopic mycelial networks that have been invisibly supporting life on Earth for hundreds of millions of years are gradually becoming visible, becoming recognized, becoming valued by human societies. Conclusion: the network of life. And so we arrive at the end of our journey through the hidden lives of fungi. We have traveled from the visible mushroom fruiting bodies to the invisible mycelial networks that form the foundation of terrestrial life. We have descended into soil, into wood, into the smallest spaces in the environment to explore the fungal communities that occupy these spaces. We have traced the evolutionary history of fungi, explored the mechanisms by which fungi sense and respond to their environment, and considered the practical benefits that fungi provide to human societies. What we have discovered is a world of profound beauty and remarkable complexity, a world that operates largely hidden from human awareness, yet a world upon which human life fundamentally depends. The mycelial networks beneath our feet form one of the largest living organisms on Earth. These networks connect plants, allowing them to exchange nutrients and information. They decompose dead organisms, recycling nutrients back into the soil. They form symbiotic relationships with countless other organisms, creating the complex webs of interdependence that we call ecosystems. Fungi have evolved over hundreds of millions of years, adapting to nearly every environment on Earth, from the depths of oceans to the tops of mountains, from deserts to Arctic tundra. They have developed remarkable capabilities, the ability to break down almost any organic compound, the ability to form complex partnerships with other organisms, the ability to sense and respond to their environment, the ability to communicate and coordinate across vast distances. Yet fungi are now facing unprecedented challenges. Climate change is altering their environments. Habitat destruction is eliminating fungal diversity at an alarming rate. Pollution threatens fungal communities in many ecosystems. Industrial agriculture is reducing fungal diversity in agricultural soils. We are in the midst of a fungal biodiversity crisis, though few people recognize it as such. Yet there is hope. Fungi are remarkably adaptable. They have survived multiple mass extinction events in Earth's history. They have co-evolved with countless other organisms, developing sophisticated relationships of interdependence. They offer potential solutions to many of humanity's problems, from remediating contaminated sites to producing sustainable materials to treating diseases. The future of fungi depends on human choices. Will we continue to destroy fungal diversity through habitat destruction and agricultural practices that reduce soil health? Or will we begin to recognize the value of fungi and work to protect and promote fungal diversity? Will we continue to overlook fungi because they are invisible? Or will we invest in fungal research and use fungal knowledge to address environmental challenges? The hidden lives of fungi, once completely unknown, are gradually being revealed. Through the work of mycologists and other scientists around the world, we are learning about the structure and function of fungal networks, about the roles fungi play in ecosystems, about the remarkable capabilities of fungi. We are learning that the fungal kingdom is not a realm of decay and disease, but rather a realm of beauty, sophistication, and life. As we conclude our exploration, we might reflect on the profound interconnectedness of all life on Earth. The trees in a forest are connected to each other through fungal networks, sharing nutrients and information. The plants in a field are supported by fungal partners that help them access water and minerals. The animals that feed on plants depend on fungi to have decomposed the organic matter that feeds the soil in which plants grow. The fungi in our own bodies play roles we are only beginning to understand. All living things are connected in a vast web of relationships, and fungi are the connective tissue of this web. In recognizing the importance of fungi, we recognize something important about the nature of life itself. Life is not a collection of independent organisms competing in isolation. Rather, life is a vast network of interdependence, where organisms support each other, communicate with each other, share resources with each other. The fungal mycelium, invisible beneath our feet, is a physical manifestation of this interconnectedness, a living network that binds the living world together. As you go forward from this exploration, we hope you will carry with you a new awareness of the fungal world that surrounds you. When you walk through a forest, recognize that you are walking on top of vast mycelial networks that are working, breaking down dead material, supporting plant growth, cycling nutrients. When you see a mushroom, recognize it as the visible manifestation of an enormous organism that may be ancient and that may be connected to countless other organisms through mycelial networks. When you tend a garden or work with soil, recognize that you are interacting with a living community of fungi and other organisms that can be supported or harmed by the practices you employ. And recognize that the hidden lives of fungi are not merely objects of scientific curiosity, but are intimately connected to your own life and well-being. The food you eat depends on fungal partners of crop plants. The medicines you take might be derived from fungi or made using fungal enzymes. The air you breathe is affected by carbon cycling that involves fungi. Your own health is influenced by the fungal communities that live on and in your body. You are not separate from the fungal kingdom, but rather a participant in the same web of life that fungi are part of. The fungal world, hidden for so long, is gradually becoming visible. As it does, we gain not only scientific knowledge, but also a deeper sense of wonder and awe at the complexity and beauty of the living world. We gain recognition of the profound interdependence of all living things. And we gain motivation to protect and cherish the living networks, visible and invisible, that support all life on Earth. Thank you for this journey through the hidden lives of fungi. May it deepen your appreciation for the extraordinary world of fungi and the roles they play in sustaining life on our planet. And may it inspire you to live in greater harmony with the fungal networks and ecosystems that sustain us all. The end.